at your location. And I welcome all the participants who have joined this webinar. So far, there are more than 100, about 125 participants have already joined. And then number is increasing gradually. I welcome all the participants, speakers, and moderator to this webinar on adapting agriculture to climate change strategies for resilience and sustainability. So I am Engineer Harish Kumar Verma, Executive Director from ICID, Central Office, New Delhi. And I welcome you, everybody. And then just a brief background on the webinar. As you are all aware, that greenhouse gas emissions and global warming have emerged as a critical climate issues in the 21st century. The relentless increase in these emissions has contributed to a rapid rise in global temperature, resulting in widespread environmental and societal consequences. In response to this pressing challenge, the target of achieving net zero emissions by 2050 has gained immense importance in the fight against climate change. And agriculture activity, they play a vital role in this climate equation, being recognized as significant source of greenhouse gases, particularly methane and nitrous oxide. These emissions result from various farming practices. It is important to note that agriculture is not solely a contributor to the problem, but also holds the potential to be part of the solution. Through the implementation of sustainable and regenerative farming techniques, the high primary production in agriculture can transform the system into a net carbon sink. Proper land management practices, rational fertilization, alternative wetting and drying, and soil carbon sequestration initiative within the agriculture sector can all contribute to offsetting emissions, offering a pathway towards achieving the coveted net zero emission target. This webinar serves as a platform to explore the intricate relationship between farming practices and the environment. By delving into innovative strategies, sustainable approaches, and the latest research findings, the webinar empowers the audience to actively engage with the complexities of climate change adaptation. And just that after the presentation, we'll have a question answer session, and all the audience may send their questions, which should be short and specific. After the written questions, if the time permits, participants will be requested to share their experience. Please indicate your willingness to share your experience by raising your hand. Two speakers for today's webinar are Dr. Cheng-I and Dr. Yo-Jin. Moderator of this webinar was Professor Ray Sean Wu. Ray Sean Wu, who is the chair of Working Group on Climate Change of ICID. Since he could not join because of his certain other engagements, so Dr. Hyo Liu will be moderating this session in his place. Now I hand over to Dr. Hyo Liu to make to move forward and then introduce the speakers and moderate the session. Thank you. Dr. Hyo Liu.
Yes. Yeah, thank you for very nice introduction of today's webinar and the content and for the two speakers. I'm Hua-Long Yu from National Taiwan University and also the Chinese Taipei Committee. And so today on behalf of Dr. Wu, because he's not available, so I will be, on behalf of him to be the moderator for today. So today, the two speakers, one is Cheng-I Xie, Professor Cheng-I Xie, and he has a very long experience in using the flux method to measure the carbon emissions from the paddy field. And also the second speaker, Yo-Jin Xiao, and he also has a very long experiences in assess the greenhouse emissions from the agricultural field. And both of them come from the National Taiwan University and Department of Bioenvironmental Systems Engineering. And so today, I think, I will hand over the talk to the first speaker. And by the way, today I will, because I may leave earlier, so I will co-chair with the Dr. Cheng-I. So he will to be the moderator afterwards. Okay, so thanks. So now I hand I think it's time for Professor Cheng-I and for his presentation.
So should I share my PowerPoint file?
Yeah, we can see your PowerPoint file. We will make it in the presentation mode. Yeah. Perfect. Yeah, it's okay.
Okay. All right. All right. Good afternoon, everyone. I'm Cheng-I. I'm from National Taiwan University. This afternoon, I will talk about energy, water vapor, and carbon dioxide fluxes above a subtropical monsoon rice paddy. Introduction. In this study, the eddy-covariance method was applied to understand the characteristics of sensible heat, water vapor, and carbon dioxide fluxes above a subtropical monsoon rice paddy in North Taiwan. The mean air temperature is 24 degrees Celsius, and the annual precipitation is around 2,500 millimeters, so it rains a lot. This is a one crop season rice paddy. The crop season is from February to June, and the fallow season is from July to December. Eddy-covariance method. For those of you who are not familiar with this method, I will have a brief introduction to this method. This method is based on the adaptation diffusion equation. So it considers the conservation of scalar mass. So if this equation is applied, then the adaptation flux, which is the second term, and the diffusion flux is balanced by the change of scalar mass and the source term. The source term is the strength that the system will release or uptake carbon dioxide or any other scalars. Here, U, V, W are the three-dimensional wind speeds. C is the concentration of the scalar. If we apply the Reynolds decomposition, then the instantaneous quantity can be divided into the mean quantity plus the fluctuation. Here, overbar denotes averaging, and the prime denotes fluctuation. So after applied this Reynolds decomposition into the adaptation diffusion equation, we end up with an additional term, which is called turbulent flux divergence term. And here, overbar again denotes averaging. So if in this equation, if we assume we have a steady state condition, so in other words, partial C bar over partial D can be ignored, and we have horizontal homogeneity condition, and the vertical mean wind speed is 0, then all these terms can be cancelled out. So we end up with a term with the flux divergence, and on the right-hand side is the source strength term. So if we do the integration, then the covariance between the vertical velocity fluctuation and the scalar fluctuation equals to the source strength plus the emission from the surface. So if we measure the turbulent flux, W prime C prime bar, then it equals to the surface source, which is the quantity that we want to know. And W prime C prime bar is the covariance of W and C. So this is why the name of eddy covariance. So for an eddy covariance system, we need a three-dimensional sonic anemometer to measure the three-dimensional wind speed and the temperature. And then also a gas analyzer to measure the water vapor, carbon dioxide, or other greenhouse gases for measuring the concentration. Then two ways can be applied. it can be open pace or closed pace to measure these concentrations. In this study, we adopted an open pace CO2 and water vapor analyzer, and this is a LICO 7500, and an IM Young sonic anemometer. The site is located in north of Taiwan, Yilan. So this is the geophysical location of the site. And again, the canopy height is from 0.1 to 1.25 meters. So this is a photo of our eddy-covariance system and above the rice paddy. So in this rice paddy, as I've said, that from February to June, this is the crop season. So this is a photo of the crop season. After the harvest, then we have the fallow season, where it's a dry land. And from July to August. Then after August, the rice paddy is flooded with water for the next crop season. So for the crop season, that the carbon dioxide flux is measured. So on this figure, we show the x-axis is the net radiation, the y-axis is the CO2 flux. So as you can see that when the net radiation is increased, the CO2 uptake by the rise is increased. However, if the net radiation is too strong, then the CO2 uptake is not increased anymore. So for the fallow season, that for from July to August, as you can see from here, there are still many vegetations on the land on the rice paddy. So you can see here when the net radiation is increased, the CO2 uptake is increased too. For the fallow season, when the rice paddy is flooded with water, so not much vegetation is left on the land. So as you can see that the CO2 uptake or release is not a function of land radiation. And here you can see that from all these three figures, that the CO2 release during nighttime is actually very small. It's around like 5 micro or more per square meter per second. From this figure, from this slide, we can see more detail about the CO2 flux. The x-axis is the Julian day, the y-axis is the CO2 flux. So you can see during daytime, the CO2 flux is negative. It means that the CO2 is uptaken by the rice. So as you can see, the maximum CO2 flux is about like minus 20 micromole per square meter per second. And during nighttime, the soil respiration is actually very small. So it's close to zero. The maximum is around, again, 5 micromole per square meter per second only. So this actually gives us a useful information is that during nighttime, the nighttime soil respiration is actually small and compared to the photosynthesis uptaken CO2. And on for this figure, it shows that the x-axis is the Julian day, the y-axis is the sensible heat flux, and the water vapor flux and the net radiation. The blue line is the net radiation. From here, you can see that the sensible heat flux is small on this rice paddy. It's because most of the energy was absorbed for evapotranspiration from the rice paddy. And as you can see that the canopy temperature and the air temperature is not that far. So that's why that the sensible heat flux is so small. And then this also give us some useful information about the rice paddy. It can provide evaporative cooling for the site. So this is this slide gives you some information about the partition of net radiation into the sensible heat flux and the latent heat flux during the crop season. So as you can see that only about 10% of the net radiation was attributed to sensible heat flux, and about 25% of the net radiation was contributed to water vapor flux, and the rest is absorbed by the rest of the energy was absorbed by the water and the ground. And in this study, we also applied Penman-Monteith equation to estimate the evapotranspiration from this rice paddy. And on this figure, is the x-axis is the Julian day, the y-axis is the major latency flux, and the Penman-Monteith equation estimated water vapor flux. You can see that the Penman-Monteith equation can reproduce the measured water vapor flux very well. And on the right hand side, you can see the one-to-one comparison between the measurements and the estimation from Penman-Monteith equation. In this study, we also study the relative turbulent transport efficiency between temperature and carbon dioxide, and the temperature and the water vapor. From this figure, the x-axis is the Bowen ratio, the y-axis is the relative transport efficiency between heat and the carbon dioxide. From here, you can see that during small Bowen ratio, for example, around like 0.15, the majority here is smaller than 1. So it means that the carbon dioxide is transported more efficient than heat. The same phenomena was noticed for water vapor too. So in water vapor, again, in Bowen ratio, in small Bowen ratio, you can see that water vapor is transported more efficient than heat. This is because carbon dioxide and water vapor transportation are both controlled by the stomata. So in conclusions, during the crop season, we have noticed that 25% of net radiation was used for latent heat flux, 10% was used for sensible heat flux, and 65% was absorbed by the water and the soil in the rice paddy. And during crop season, the maximum CO2 uptake was about 22 micromole per square meter per second. In other words, this is about 35 kilogram per hectare per hour. So if there's one day the daytime is around 8 hours, then if you multiply this by 8 hours, you will get the CO2 uptake per day. So in the fallow period, the maximum CO2 emission rate from the soil water service was about only 5 micromole per square meter per second. Remember, this is the maximum. Normally, it's close to 0. So this 5 micromole per square meter per second is about 8 kilogram per hectare per hour. And also, we found out that Penman-Monteith equation was found to reproduce the evapotranspiration well with surface resistance close to 190 seconds per meter. This value is a little bit bigger than the value that we experienced. This is maybe that when we are doing the Penman-Monteith equation estimation, we did not take into account the energy balance ratio into account. And also, we found out that under small Bowen ratio conditions, water vapor and carbon dioxide were transported more efficiently than heat. That's all about my talk. Thank you very much. So, any questions? Oh. Hello?
We will take all the questions at the end of the presentations, okay?
Okay.
Well, we have requested all the participants, those who are interested in having any questions, that they should send the questions to question and answer box. And then those questions will be answered by the respective speakers. All the attendees are requested to share their questions as and when they have any question. Thank you.
You can also refer to Q&A button to see the questions. Okay.
Dr. Yo-Jin.
Yes, can you hear me?
Yeah.
Okay, so should I start my presentation right now?
Yeah, please continue.
Okay, let me check. Let me share my screen first. Okay, I don't know if you can see my slides right now. Well, okay. So it seems like everything looks fine. I assume. Okay. Good afternoon, everyone. It's my honor to have a chance to share some of the work that we did in the past few years with you about the topic I'm going to share today, which is the adapting agriculture strategy for achieving net zero greenhouse gas emission goals. And for today's presentation, I think the slides I prepared may be a little bit too basic for you. So I apologize for that if you think the information are not informative for you. Okay. So I just want to give you a basic introduction about myself because I'm actually a biogeochemist and also environmental microbiologist. So the presentation I'm going to introduce you about this topic would be from more like a microbial scale or from a biogeochemical process scale instead of from field scale. Okay. And so in conclusion, okay, so what I'm going to talk today, it will lead to a conclusion here. Because for this today's presentation, I think for those of you who came into this presentation, you may assume that we will get a conclusion about how we can attain a net zero greenhouse gas emission goal for agricultural ecosystem. But the conclusion for this answer is that we do not have a clear answer to this question. Okay, so my today's presentation, I probably would not be able to give you a clear answer for this question. I would probably able to share some of the perspective and some of the results that we found so far that may help us to find the answer in the future for this topic. Okay, so what global warming is, I think all of you or probably majority of you may already know about this, but the basic idea is that the global warming lay on the greenhouse gas effect. So it means some of the gas in the atmosphere can absorb some of the radiation from the reflection refraction from the Earth, and then turn that into heat. So the most commonly known greenhouse gas, including the carbon dioxide, methane, and nitrous oxide. All of those greenhouse gas can trap heat and warming the plants and maintain a temperature that suitable for life on Earth. The problem is that because of the human activity, these human activity may be from like burning fossil fuel, deforestation, or different kind of industrial process that increase the concentration of the greenhouse gas I mentioned earlier. And these assessed greenhouse gas may stimulate or may increase the global warming effect to increase the atmosphere temperature on Earth. So as you can see from the graph here, the IPCC report shows that the concentration, the greenhouse gas concentration almost doubled in the past 50 years. And because of these enhanced greenhouse gases, it leads to an increase of global temperatures. And depend on different trajectory, the increment of atmosphere temperature is projected to be more than 1.5 degrees Celsius in the end of 2100. And in order to stay the atmosphere temperature below 1.5 degrees Celsius, the IPCC suggests that all countries around the world need to reduce the greenhouse gas emission and increase the carbon removal from the atmosphere, so that we will be able to, we need to do that in 2050, so that we will be able to reduce the global temperature, the increment of temperature below 1.5 degrees Celsius. And the way to achieve that net zero emission rely on different methods. One method believed to have high potential for removing carbon dioxide from atmosphere is the natural carbon sinks. Okay, so natural carbon sinks are considered the most cost-effective solution for achieving the 2050 net zero goals. The concept of natural carbon sink relies on high primary production in some natural ecosystems, such as the forest and wetlands, and for the grasslands. And cropland may also be an important ecosystem with high potential of high potential primary production that may remove carbon dioxide from the atmosphere. Just like what you listened earlier from Dr. Xie, he present the way to use the eddy-covariance method to measure how much carbon dioxide can be sequestrated by crop by the rice paddy crops. But at the same time, natural ecosystem sometimes can also be important greenhouse gas sources, especially for nitrous oxide or for the methane, and depends on different type of agricultural practice. At a certain point, or for some practice, nitrous oxide and methane can be produced. And all of these greenhouse gas are produced because of mostly produced because of the soil microbial activities. So here is a very simple graph, shows the idea about how microbes living in soil environment may produce the greenhouse gas. So imagine here is a soil microbe. When it want to live, it need to eat some food. Okay, so on the left-hand side, it shows the organic matter, which is the food for the microbe. So when microbe take or eat the organic matter, it release nitrogen and proton. And these nitrogen and proton cannot be stay in the soil environment alone. It need to be absorbed by some of the chemical compound and to form a chemical molecular. So under aerobic environment, those proton and electron can be absorbed by by oxygen and generate the water molecular. And during this process, it releases some of the carbon dioxide to the atmosphere. However, when there is no oxygen in the soil environment, which mostly happen in an aerobic environment, which means when water covers the soil and the oxygen cannot absorb into the soil pore space, then at that time there was no oxygen can be utilized by soil microbe. So that the soil microbe need to find some alternative chemical compound to absorb the proton and the electron. And the alternative chemical compound that microbe use can be nitrate. Okay, and when the microbe use nitrate as an electron acceptor and proton acceptor, then this nitrate can be reduced into nitrous oxide, which is one major greenhouse gas that can be produced in agricultural ecosystem. And this nitrous oxide can be very important to regulate the 2050 net zero emission, especially for agricultural ecosystem, because this nitrous oxide for molecular or per mole of nitrous oxide, it can be 298 times more efficient than the carbon dioxide in tripping heat. So for per mole of nitrous oxide, it will have equivalent effect in tripping heat as the 298 mole of carbon dioxide. And under another environmental condition, soil microbe may use carbon dioxide as electron acceptor. And under this situation, the carbon dioxide can transform into methane. And this methane is another important greenhouse gas. And it has 25 times more efficient in tripping heat than the carbon dioxide. So that under agricultural environment, even though some of the plants can absorb carbon dioxide due to the photosynthesis, but some of the soil microbe may release the methane and nitrous oxide back into the atmosphere and offset the effect of plants' uptake of the carbon dioxide. So in this way, when consider whether or not agricultural ecosystem can be an effective carbon sink, we need to do what we can do is to do a simple mass balance. So for aerobic environment, if we want to do a mass balance to determine whether or not the carbon dioxide can be stored in this environment, what we can do is either we measure the amount of mass into this system or amount of carbon dioxide that's uptake by the plants into this system, and how much of the carbon dioxide released back into the atmosphere, which can be determined by the method such as eddy-covariance method that Dr. Xie present earlier. Or what we can do is we can simply measure how much of the carbon dioxide or how much of carbon stored in these systems by measuring the plant carbon and the soil carbon. Okay, because the equation is equal, so whether or not you can determine the left-hand side or you can determine the value from the right-hand side. Okay, as long as this equation can be larger than 0, it means this ecosystem can sequestrate some of the carbon dioxide from the atmosphere. Okay, so for example, if we measure 3 units or 3 more of carbon dioxide from the atmosphere and release 2 more of carbon dioxide back into the atmosphere, we know that 1 more of carbon dioxide is stored in this system. The problem is for the anaerobic environment, because under this environment, some of the carbon can release back into the atmosphere as methane. And some of the nitrous oxide may be produced if there is nitrogen source in the soil environment. Okay, so the equation this balance can also be calculated by mass balance equation. The problem is this: we not only need to consider the amount of methane emission from this system, but also we need to determine how much of nitrous oxide released from this type of anaerobic environment. So the basic equation, just like the equation from the aerobic environment, we can determine the amount of plant carbon, amount of soil carbon, or we can determine how much of carbon dioxide come into this ecosystem and how much of carbon dioxide and methane released back to the atmosphere. As long as this equation equal to 0, this means the carbon balance, and there is more carbon dioxide coming into the system than the amount of carbon released back into the atmosphere. The problem is not only we need to know how there are more carbon dioxide absorbed in this system, but also we need to know the amount of carbon dioxide that's sequestrated in this system can have a positive effect to reduce the global warming potential. So that we also need to know the amount of carbon dioxide that's sequestrated by the plants and soil need to be higher than the amount of carbon dioxide that released back into the atmosphere. But at the same time, we need to know that it need to be 25 times more than the amount of methane released back into the atmosphere. And also the amount of carbon dioxide need to be more or higher than the amount of 20 or almost 300 times higher than the amount of nitrous oxide released back into the atmosphere. So that we can guarantee under this situation, this type of ecosystem may have a positive effect to reduce the global warming potential. The problem is for the agricultural ecosystem, you can see from here in the purple bar or purple sign here. Agriculture is believed to be important source to methane and nitrous oxide, especially for rice paddy, because it's an anaerobic environment, so that soil microbe can only do the anaerobic respiration, release some of the methane and the nitrous oxide. And because of that, and also because of the over-fertilization, agricultural activity is believed to produce lots of nitrous oxide, and those nitrous oxide are contributed by the chemical fertilizer. And this chemical fertilizer stimulates soil microbial activity and then facilitate the nitrifying and denitrifying process in farmland. And at the same time, nitrifying and denitrifying process also release some of the what do we say, nitrogen species gases that affect different or cause different air pollutions. But those nitrogen species gas such as HONO, such as nitrate oxide, are not the topic we are going to talk about today. What we are going to focus on is the nitrous oxide. Okay, so what we did in 2019, we started an experiment in Taichung, Taiwan, and we determined different nitrogen species gas and soil microbial composition in paddy, in rice paddy, and also in Chinese cabbage plantation. And this is the situation of the site. We also use the eddy-covariance equipment to measure the greenhouse gas emission and carbon dioxide emission. At the same time, we also measure the nitrous oxide using the eddy-covariance method in the site. Okay, and for the fertilizer, we put in the Chinese cabbage and the rice paddy soil are listed here. Okay. At the same time, we also collect the soil sample because we know that these greenhouse gas may when these greenhouse gas may be produced by the soil microbes, so that we also collect soil sample in 3 different plots. Okay, for each plot we collect 15 soil core to analyze the soil chemical process and the property and the soil microbial property for those soil in different times. Okay, so the for the soil samples, we analyze the soil total organic carbon. We analyze different cation and anion concentration in the soil. We analyze the pH, EC, and the soil temperature, water content, and we analyze the soil microbial community using the Illumina MySeq and quantitative PCR. So this is the results about our experiments. And the graph shown here is the Chinese cabbage practice in 2020s. So these experiments, this experiments was done in past 3 years from 2019, 2020, and 2021. but because of time, I just show you some of the results here as an example. So as you can see from here, the cabbage practice in 2020, the accumulated nitrous oxide concentration was about 740 kilogram of the carbon dioxide equivalent. So this means the amount of nitrous oxide was multiplied by 298 to get the carbon dioxide equivalent value. And for the carbon dioxide, the amount of carbon dioxide that is sequestered by the plants and by this ecosystem was about negative 697 kilogram of carbon dioxide. So this means when these two, the amount of carbon dioxide equivalent that attributed by the nitrous oxide it is actually higher than the amount of carbon dioxide that's sequestered by the plants in our farmlands. So that the farm, this farm actually acts as a carbon dioxide source or greenhouse gas source than a greenhouse gas sink. Okay, and the graph shows on the right hand side indicate the time difference about the nitrous oxide emission and carbon dioxide emission in hour per hour. And when we determine how much of the nitrous oxide emission in relate to the nitrogen input or in relate to the fertilizer input, we actually found for per unit of nitrogen input into the farmlands, there was about 2 to 2.5% of the emission factor. So it means for maybe for 1 kilogram of nitrogen put into the farmland, there will be 0.2 to 0.25 kilogram of nitrous oxide be generated from these farmlands. Okay, so the emission factor was about 2 to 2.5. And when we look into the pattern of nitrous oxide emission, we actually found that most of the nitrous oxide emission was found just after the fertilization. So as you can see here, the yellow bar, it shows when the time that fertilizer was applied into the farmlands. And not only that, we also found actually there was higher nitrous oxide emission from the cabbage plantation than the rice paddy plantation. So so you can see from here the highest nitrous oxide emission in the Chinese cabbage soil was actually way higher than the amount of nitrous oxide emission from the rice paddy. Here you can see the units are actually different, showing on the y-axis. Okay, so we suspect that the nitrous oxide might be due to the nitrification process, because from literature, most of the literature says that in a dry farmlands, the nitrous oxide emissions is actually due to the soil nitrification process. Sorry, but and this this is the information about the soil physiochemical properties. They're showing no significant of soil total organic carbon change during the Chinese cabbage plantations. But when we look into the soil microbial composition, we actually found that the nitrification process, okay, or the nitrification or nitrifying microbial communities were actually not significantly higher than the denitrifier in Chinese cabbage plantation. So actually, at some point, actually denitrifier were found higher than nitrifier in some period in Chinese cabbage plantations. So this provides some of the contradiction to the nitrous oxide emission we observe in the farmlands. And so we continue to do different analysis. So, and we found out that microbial metabolic pathway in Chinese cabbage farmland and also in rice paddy farmland actually shows that majority of nitrogen cycle were denitrification process. Nitrification process was the minor process in both rice paddy and Chinese cabbage farmlands. And we also did some of the statistical analysis, and we found out that in an environment with intermediate water, soil water content, it may stimulate the incomplete denitrification process. This process may stimulate the amount of nitrous oxide emission from the farmland to the atmosphere. But we are still curious about why we cannot find lots of nitrifier in the dry land. So we also conduct the nitrification activity using an ethylene inhibition method. This method inhibit the nitrification and also inhibit the N2O emission in the not N2O emission, inhibit the N2O reduction to the nitrogen gas in the denitrification process. By using this ethylene inhibition method, we will be able to determine how much nitrous oxide are produced by the denitrification process and the nitrification process in the farmlands. And actually, I think because of time, by using the ethylene inhibition method, when the soil incubate by the ethylene, we should be able to see lots of ammonium increase in the soil 2 times. But without the ethylene, we should be able to see lots of nitrate accumulate in the soil 2 times. So this graph actually shows the inhibition methods. The purple and the orange bar shows on the most right hand side is the sample the soil sample inhibit with ethylene. So, but from the result we can see here, the ammonium concentration did not significantly increase with ethylene inhibition. This means that the soil may have very weak nitrification process in our farmlands. Okay, and we also conduct some of the statistical analysis to determine why that the nitrification process was not occur in our farmlands, and then we found some of the positive and negative correlation of the soil physiochemical properties with the nitrifying communities, microbial communities. So that we have a quick summary for our experiments. So we actually found that nitrification activity was low in both plantations, farms, and no N2O flux was observed in the incubation experiments. So it means the N2O emission from the farmlands were actually or were most likely from the denitrification process. Okay. And intermediate, we also found the intermediate soil water content may stimulate incomplete denitrification in farmland and contributing to the N2O emission. And we also found the over fertilization also contribute the N2O emission from the farmland, reduce the carbon negative aspect of agricultural ecosystems. So this comes to a question, which is how can the proper for our future study, which is the how can we manage a proper irrigation strategy to not stimulate the incomplete denitrification. And this would be one of the questions we need to answer in the future. And there's also one question we need to keep finding the answer, which is how can one design a reasonable fertilization plan to minimize N2O efflux? Because all of these rely on proper drainage and irrigation strategy to maybe optimize the water level, reduce nutrient runoff, and in that way, we will be able to limit the greenhouse gas emission from agriculture ecosystem and making this system to be a net carbon sink. Okay, so this is basic the presentation I have today for you, and thank you very much for your attention. And I think this time for taking questions.
Thank you, Dr. Yo, and thank you, Dr. Cheng, for this interesting presentation. So now we have our President Honorary Professor Raghav and President Honorary Dr. Peter Lee. So, Dr. Raghav, you have few questions. So you can give your remarks also, and then you can ask questions from the speakers and get answers from them.
Okay, thank you, Engineer Varma. My first question is related to the eddy-covariance. And eddy-covariance, the first speaker referred to the fact that he used 190 second per meter in order to get the Penman-Monteith equation values to be closer to the measured eddy-covariance. I think it is quite high compared to what was suggested by FAO 56. The modified Penman-Monteith equation by FAO 56, they suggested 70 second per meter, and that led to overestimation of evaporation by several scientists. And there is no wonder why actually the first speaker found a higher value, like 190 second per meter, as canopy resistance to make match between the eddy-covariance and the Penman-Monteith evaporation. I would like to have some elaboration on that, and also I would like to see if that work is published, and if it's published, I would be happy to have a look at the reference and the paper. That's for the first speaker. Second speaker, the denitrification that he referred to, of course, it is the main issue with producing methane gas in paddy rice cultivation. Why don't we move towards dry rice cultivation, and we just avoid using paddy rice cultivation and move to a dry rice? In India, for example, now they started to use drip irrigation for rice cultivation. Australia, they started to use dry rice cultivation. And in order to solve this issue of really gas emission from paddy rice, I think we should stop using that way of irrigation, and we move towards dry rice cultivation. That's just an observation and a comment to the question, because the second speaker was asking, how can we solve the problem of methane production in paddy rice? The answer is, don't actually use that sort of way of irrigation and cultivation of rice. Simply go to dry rice cultivation. Thank you.
Dr. Cheng and Dr. Yo, you can respond to the sessions and questions from
yes. Okay, I will answer about the canopy resistance, about 190. Yes, I agree with him that this value is a little bit higher than FAO the FL, this 190 is the canopy resistance for actual evapotranspiration. But again, I also agree that this value is a little bit higher, but we try to mimic the measurements. So this is the value that we get, and I think probably some of the problem is attributed to the energy closure method, because the energy closure problem, because at this site we do not measure the heat storage in the water. So actually we have to force that the rest of the energy is actually the energy stored in the water and the soil. And this is the first question. And the second question is about if this work is published. Yes, we have this part of it's published. We recently published a paper in Water regarding using machine learning approaches to estimate carbon dioxide flux and water vapor flux in this rice paddy field. So I'm more than happy to provide a copy to you. Thank you very much for your question.
Okay, you can put it in the chat box if you the title of the paper. You can put it in the chat box, and then we can copy that reference from there.
Okay.
If you...
Yeah. Okay. Thank you. Yeah, I will do now.
Dr. Yo.
Okay, thank you for the question. Sorry for not present as clear as I supposed to. In the very end of my my presentation was actually about the nitrous oxide N2O, not the methane CH4. Because from the research we found out that N2O emission may mostly occur when the soil is more aerobic. So theoretically, and actually what we are currently working on the continuing project is we are also considering using different kind of irrigation method. We are not culture the rice as the way we used to do, just like what you mentioned earlier about using drip method or use dry rice cultivation. The problem is that when the soil is getting more aerobic, the N2O emission can be getting higher. Even though this dry rice cultivation or this kind of method may reduce the amount of CH4 emission, but at certain points, we may probably reduce the CH4 emission, but increase the N2O emission. And this is something we don't really want to see, because the N2O emission can be 298 times higher in tripping heat than carbon dioxide. But the methane, the CH4, can be 25 times higher than carbon dioxide. So if we reduce the amount of CH4 emission, but increase at the same time increase the N2O emission, we will actually increase the overall greenhouse gas emission for the farmland ecosystem. So that but this N2O emission, even though it tend to happen in more aerobic soil environment, but there seems to be a kind of a threshold or a certain point. So under some kind of soil moisture, this N2O may not emit into the soil. But if the soil getting too dry, then this N2O can be released even more than different kind of ecosystem. So this is something we need to evaluate to see how much soil water content can be to reduce the CH4 but not increase the N2O. And that's something we still not find the answer. Thank you.
Can I comment further on that?
Yeah,
Please.
Dr. Raga.
Well, I think the issue is really methane or nitrous oxide emission. I'm talking generally about the gases emission from paddy rice. And when you create an aerobic condition, is it because of the fact that you are flooding the fields? When you flood the fields there, then you create anaerobic conditions and you create that gas emission and so on. And also you create a mosquito environment and of course malaria as well. So there are a lot of really environmental and health issues related to flooded type of irrigation and creating anaerobic conditions. You can avoid anaerobic conditions by cultivating rice in a way that the Australians do, and also some parts in India now, they started to use drip irrigation, and there is no anaerobic condition under dry rice cultivation. I think we should not encourage having to cultivate rice as a paddy rice, and we should really move away from really flooding the fields and wasting a lot of water, wasting fertilizers, polluting groundwater, a lot of really environmental and negative impacts. And we shouldn't really study and put a lot of money investigation into studying a system that should not exist. And we should move away from paddy rice. That's really my opinion.
Thank you very much. I totally agree. I totally agree. Actually, we are currently working on we are also working on the dripping irrigation right now, and we want to determine how frequent we need to irrigate the rice with the dripping method to reduce, not only reduce the CH4 but also inhibit the amount of N2O emission, so that we may hopefully we can find maybe a happy point for the dripping irrigation. But I totally agree that we should give up the traditional rice paddy, the traditional irrigation for rice paddy, and turn that into the dripping method. That's things I totally agree with, and that's also the experiment that we are currently working on. Thank you.
And one thing I would like to add, most of the climate change models are developed either in Europe or in America, and they don't work in Asia for one reason, because all the Americans or European models, they depend on the CO2 only, while in Asia you have more different type of gas emission like CO2 or methane, and that's why these climate change models that developed in America or in Europe, they don't really apply correctly in Asia because of the paddy rice areas that really dominates most of Asian land. Thank you.
Thank you, Professor Raghav, for your remarks as well as suggestions and then comments. Now we have a question. Number of questions were raised in Q&A box, which were replied by both the authors. So one question is for us that whether recording will be available. Yes, recording will be available. In a day or two, we'll process the recording. A recording will be available on our website. So then anybody can go through the recording and they can download also. So that is one thing. And regarding presentation, if authors agree, then we can share the presentation in PDF form. So once they confirm, then we'll put up the presentation also on our website along with the recording. And Mr. Morgan Cole, he has raised hand. Now we are giving this speaking rights to him. Then he can unmute and then speak and raise his question. Mr. Morgan Cole, you can unmute yourself.
Sorry, yes?
Oh, sorry. Yeah. Ms. Morgan Cole, you can now unmute and then you can raise your question because you have raised the hand. If you have any any suggestion, any comment, any question, then you can name the speaker and then you can tell your question.
Perfect. Sorry, that was on mistake. Thanks for checking though.
Thank you. So if anybody has any questions, then they can raise the hand. Otherwise, if there are no questions, then I thank both the speakers, Dr. Chen and Dr. Yo, and as well as Dr. Fallow, who has acted as a moderator. We also thank Professor Raga Raghav, our President Honorary, and President Honorary Peter Lee, who were also a part of this webinar. And I thank all the participants who have joined this webinar. The total number was around 150. So this is a good number as compared to the webinars which we are organizing. So thank you very much to all the participants, all the speakers, all the attendees.